Okay, welcome to cellular respiration. This chapter is extremely dense, and I really recommend that you watch the accompanied YouTube video that I posted regarding the processes involved in cellular respiration. This week we're going to be focusing on glycolysis, which is anaerobic, and next week we will be going into the aerobic processes of the Krebs cycle and the electron transport chain. So we've talked a lot about energy in the last few weeks. Energy is this coordination of chemical pathways. It's a combination of oxidation and reduction reactions. So oxidation is when an electron is stripped from an atom in a compound, and reduction is when an electron is added to an atom in a compound. These generally do not occur on their own. They usually occur together at one time, and that is why they are called redox reactions. So once an electron is stripped off of one atom, it's generally added immediately to another atom in that compound. So the oxidation of a compound lowers its potential energy, and when that removed electron from the oxidation of that compound moves to another compound reducing it, it increases its potential energy. So it's this constant flux of lowering potential energy in one thing, increasing potential energy in another. So energy stored in atoms and used to fuel cell function is in the form of these high energy electrons. And this allows there to be incremental changes in energy rather than large bursts of energy. So how are these electrons transferred? How are they carried? Electron carriers. So they bind and carry these high energy electrons between compounds. They're kind of like little shuttle systems. Energy or electron carriers can be easily reduced or oxidized and tend to be derivatives of B vitamins. So this is one major reason why B vitamins are so important in the body. Without your B vitamins, and there are many reasons why they can be deficient, especially if you're taking certain medications like oral contraceptives. It is super important to supplement your body with B vitamins so that you're able to make and use energy. So NAD is one of our electron carriers. It's a derivative of vitamin B3. NAD plus is the oxidized form, NADH is the reduced form. FAD plus is a derivative of riboflavin. The reduced form of that is FADH2. So you might notice that the reduced forms tend to have H's in them, so that might be a way to remember it. Both NAD and FAD are used in the energy extraction from sugars, and NAD plus, which is NAD plus a phosphate group, is involved with anabolic reactions and photosynthesis. Living cells cannot store much free energy, and that's because it would increase the heat in the cell. If there was a bunch of energy just bouncing around in a cell, there would be a lot of heat, which would cause damage and eventually destroy the cell. That's why it's important that ATP is used as our energy currency of cells. ATP can be broken down into ADP plus a phosphate group, and that dephosphorylation and that breakage of that phosphoanhydride bond is what creates the energy that we harness for these chemical processes in our bodies. So at its core, ATP contains AMP, which is adenine plus ribose, a sugar, and one phosphate group. So that stands for adenosine monophosphate. Monophosphate, one phosphate. An addition of two more phosphate groups would be adenosine triphosphate. Both ADP and ATP are relatively unstable because they have those phosphate groups. Phosphate groups tend to have negative charges, so they repel each other. Phosphorylation is a process that is going to require energy, and dephosphorylation is a process that will release energy. So dephosphorylation is the removal of these one or more phosphate groups. So the hydrolysis of ATP into ADP plus a phosphate group is what gives us our energy. energy is needed to regenerate ATP. So it's kind of a cycle. We need energy to create energy. And this energy is going to come from the metabolism of glucose, fructose, or galactose. So you can't make this energy in your body unless you're taking in energy from other organisms like plants or animals. So phosphorylation, we said, is the binding of another phosphate group to a compound, and that requires energy. So enzymes bind to a substrate forming an intermediate. ATP will form an intermediate complex with a substrate and an enzyme, allowing that ATP to transfer a phosphate group to its substrate. When that intermediate breaks apart, the energy modifies the substrate and converts it into the product ADP plus the phosphate group. So this is just a really complex way of describing how ATP is dephosphorylated. In substrate phosphorylation, ATP molecules can be generated by the catabolism of glucose. So this is very specific, very direct, and we're directly taking that phosphate group and adding it to ADP to make ATP. With oxidative phosphorylation, this is where most of our ATP is generated from through glucose metabolism. It's a process called chemiosmosis. This occurs in your mitochondria, generates about 90% of the ATP that your body uses. It's also used in light reactions in photosynthesis, which we'll talk about in a few weeks. And chemiosmosis is considered oxidative phosphorylation because the process as a whole requires oxygen. So our first step in the cellular respiration process is glycolysis. Glycolysis is the breakdown of glucose. Simple as that. Glycolysis is an anaerobic process, so it does not require oxygen in order for it to occur. It takes place in the cytoplasm in both prokaryotes and eukaryotes. It begins with glucose and ends in pyruvate. Pyruvate will be a product that we use in our next step in cellular respiration, the Krebs cycle. There are 10 steps in glycolysis. The first 5 require energy. The last 5 make energy. And glucose will enter cells to start this process of glycolysis in the cytoplasm via secondary active transport or through GLUT proteins, which are a carrier protein that uses facilitated diffusion. So we're going to go through each of these 10 steps. You should know the reactant, the product, the enzyme that gets you there, and any of the specific things that are happening at these steps. So remember, we said the first 5 steps use energy. So glucose we take in as a simple sugar from our food that we eat. Glucose will be converted to glucose 6-phosphate, usually abbreviated G6P. It's converted into G6P by the enzyme hexokinase. So anytime you see kinase as a type of enzyme, that's telling you that we're adding a phosphate group. So hexokinase phosphorylates glucose using ATP. So we're taking that ATP, that phosphate group off of the ATP to convert it to ADP and using it to phosphorylate that glucose into G6P. Once it's G6P, that glucose cannot leave the cell anymore because it has that negatively charged phosphate group on it. It also cannot interact with GLUT proteins, so it cannot be transported in that fashion either. It's now more reactive as well because of that phosphate group. In the second step, G6P becomes F6P, or fructose 6-phosphate. Fructose 6-phosphate is an isomer of glucose 6-phosphate. As you can see, their chemical structures are very similar. So the enzyme used to create this is phosphoglucose isomerase because it's creating an isomer. In the third step, we're going to phosphorylate F6P into F16BP, or fructose 1,6 bisphosphate. Bisphosphate means that there's two phosphates. And because there are two numbers, that also tells you that there's two of whatever follows those numbers. Those numbers tell you which carbons those phosphate groups are attached to. So in this case, carbons 1 and 6. So the phosphorylation of F6P into F16BP is catalyzed by this enzyme phosphofructokinase, usually called PFK. PFK is super important because it's our first rate-limiting enzyme. So being a rate-limiting enzyme means that this process cannot happen unless PFK is in its specific conditions. PFK is active when ADP levels are high and when ATP levels are low, because this whole process is going to make ATP. So it will know if there is a surplus of ADP, we can make more ATP. This is also another step that's going to use the phosphate group from ATP to phosphorylate its product. In steps 4 and 5, F16BP is going to be converted into two different products. So we have the enzyme aldolase, and it will cleave F16BP into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So dihydroxyacetone phosphate is usually abbreviated DAP, and glyceraldehyde 3-phosphate is usually G3P. Dihydroxyacetone phosphate will then transform into G3P via an isomerase because they are isomers. So although both of these products are made through the cleavage of this F16BP, more of G3P will be made as DAP is transformed into G3P. So that's the first 5 steps. The first 5 steps are the steps that are going to use energy. So at this point, we've used 2 ATP to break down 1 glucose molecule. So remember, glucose was transformed into G6P, which was transformed into F6P, which was transformed into F16BP, which was cleaved into DAP and G3P. Those are the first 5 steps, and here are the 2 steps, 1 and 3, that use ATP to phosphorylate their products. Moving into the second half of glycolysis. So remember, DAP is going to be transformed into G3P, so we'll have way more of that. G3P will then be oxidized. It will be oxidized and phosphorylated. So G3P will become 1,3-bisphosphoglycerate, or 1,3-BPG. And again, because it's bisphospho, we know there are 2 phosphate groups. 1 and 3 tell us which carbons, so at 1, carbon 1 and carbon 3. In this case, we're not using a phosphate group from ATP. We're actually using it through the high energy electrons of NAD. The enzyme that we're going to use is glyceraldehyde 3-phosphate dehydrogenase. And you can also remember that anytime you use a dehydrogenase, its coenzyme is going to be NAD. Step 6 is also a rate-limiting step because it depends on NAD being present. In step 7, so just be careful of these pictures now because the arrows are moving to the left. Our 1,3-bisphosphoglycerate, this is our first step that's actually going to make ATP. It's going to donate a phosphate group to ADP to form 3-phosphoglycerate. So it lost its phosphate group at carbon 1. The enzyme that we're going to use here is phosphoglycerate kinase. So remember again, kinases are when there is a phosphorylation or dephosphorylation event. We lost a phosphate group, so that makes this a kinase. So we formed our first molecule of ATP in step 7. In step 8, the phosphate group will move from the 3rd carbon to the 2nd carbon. So 3-phosphoglycerate becomes 2-phosphoglycerate via enzyme phosphoglycerate mutase. So it just jumps from carbon 3 to carbon 2. In step 9, 2-phosphoglycerate will become phosphoenol pyruvate, or PEP, through the enzyme enolase. We will also lose water at this step as a byproduct. PEP is also a very high energy compound. And in step 10, our last step of glycolysis, PEP will become pyruvate via pyruvate kinase. So again, this is a dephosphorylation event. We're taking that phosphate group from PEP and donating it to ADP to make our second ATP molecule. This is also a rate-limiting step because it relies on the presence of pyruvate kinase to be in its optimal conditions. So as a recap of our second half of glycolysis, we made 2 molecules of ATP. Glyceraldehyde 3-phosphate turned into 1,3-bisphosphoglycerate to 3-phosphoglycerate, 2-phosphoglycerate, PEP, and then pyruvate. So you can see here that this step actually happens twice. So steps 6 through 10 will go through twice. So at the end of this, we are actually making 4 ATP molecules. So we're beginning with glucose, and we're going to make 2 pyruvate molecules, 4 ATP, and 2 NADH. However, we used 2 ATP at the beginning, so we netted 2 ATP from that entire process. For some cells, glycolysis is the only source of energy. For red blood cells, they don't have mitochondria. So because this process is happening in the cytoplasm, that's how they receive their energy. Without glycolysis for red blood cells, they would die. So next week we'll talk about aerobic respiration, the Krebs cycle, the electron transport chain, and so on.